Okay, let's get started. We were trying to solve a problem, and that was to get through all these checkpoints. And our poster child of the checkpoint is going to be trying to get right through here. Okay, so this is what we need is two things. We need an activated, fully activated mitotic CDK. So that it's got a cyclin, it's got to have the kinase, and then we must have completed all the events downstream from the previous checkpoint. So one of the big ones is DNA replication. We have to duplicate the MPOC, that is the centrosome, so there has to be two of those, there'll be two poles. And so that's the stuff that this thing that would cause the inhibition of this checkpoint is if these things are not completed. Okay, so and we'll talk about exactly what happens when things go wrong. Okay. All right, so we were talking about this. That's what I can't. This is the engine, cell cycle engine, and we're really are going to use a car analogy because what we talked about, this CDK is not good without its cyclin. So it binds and it actually depresses this inhibitory threonine T loop. Okay, so now it's partially activated, but then, and really in order to put your foot on the accelerator, in this case, this phosphate at a particularly conserved threonine residue is your accelerator. So if you add that one phosphate, this goes up, the activity goes up 300-fold. Okay. But that's actually not, that hasn't happened. But what actually happens first is before any of this happens, we have another player in this sequence that's actually inhibitory. So you can think of this little guy here, and he is a little guy, E1, okay, as the clutch. So I always ask people, how many people can drive the standard? Okay. So when before you shift gears, what do you have to do? You got your clutch in, you shift it to the gear that you want, let's say first gear. Okay. And then what do? You let off the clutch and then you engage the gas. Okay. What happens if you have the clutch in and you push on the gas? The RPMs in the engine go way up, right? But you don't go anywhere because the clutch has not released the drive mechanism to go forward. That's what's called a transmission. It transmits that energy to the drive shaft. Okay, so what happens here is this puts this thing in the clutch. So it's weakly activated because it's bound to that cyclin. So it's sort of, 1,000 RPM. You're not going anywhere with 1,000. And so what happens is it then adds a phosphate to a very conserved, not threonine, not serine, but to a tyrosine. This is kind of weird, putting phosphorylating a tyrosine on this protein that's floating around. Usually you reserve phosphotyrosine for what kind of protein? Signaling protein, right? Okay. Well, this is a negative. This is a clutch that inhibits this entire process. Okay. And so even if you come back and then CAT activate it, because that's what happens next. CAT goes ahead and puts his phosphate on the accelerator. So this thing is sitting there with the clutch in, revving the engine. That's what it's doing. So the key to getting past the checkpoint is 1/3 protein, okay? And it's this one right here. This is what pops the clutch. This is a tyrosine phosphatase. It's called CDC25, so it came from what animal? It's not an animal, it's a different, it's a yeast, okay? Cell division cycle is what that stands for. Okay, and a lot of these proteins were first isolated in yeast, and so you'll see actually the very first CDK, which is CDK. This is CDK1, and this is cyclin B. Those are the two specific names you need to remember. This is the generic term, and so that's the only one you really need to know, but we'll run into others today. Okay, so this puts the clutch in, this puts your foot on the gas, and so it's just revving, but it's not going anywhere. And then this thing needs to be phosphorylated, so it gets phosphorylated probably because this is weakly active, and when this is phosphorylated, it becomes active, and it binds and it clips off that tyrosine phosphate, and that lets the clutch out, and this thing roars forward because there's a positive feedback loop. And the reason it's a positive feedback loop is that once this is fully active, it shifts into here, it's moving down the highway, right? One of the first proteins that phosphorylates is CDC25, which then activates more, which then phosphorylates more, so that you have a positive feedback, and so the activity goes through the roof. Okay? And then the second part of the positive feedback is that this active CDK phosphorylates WEE1 and inhibits its action. So the positive feedback is forward, making more phosphorylations onto this, making that active, and also inhibiting the inhibitor. Okay. Now, why is it called WEE1? If you mutate this protein, first of all, you start to speak Irish and call things by Irish names. And so what happens is, if you inhibit this protein, the cells are very small. Think about why that would be the case. What if you don't have WEE1? What happens? So as soon as this gets made, this is going to be unopposed. And so are you going to go through the checkpoint or not go through the checkpoint? You're going to go very fast. And so you skip G2. And so there's no gap between DNA synthesis and mitosis. That's what a cancer cell does. That is a cancer mutation. What kind of protein is this? Is this an oncogene or is it a tumor suppressor? It's a tumor suppressor, right? Because if it's active and you don't inactivate it, it suppresses the ability of that tumor cell to go through mitosis. Right? So the way you always answer that question is, if I make a lot of it and it's active, does it cause mitosis or inhibit mitosis? That's the answer to that question tells you whether it's a tumor suppressor or not. So let's try it with this one. If there's a lot of that protein, so much that it outcompetes this guy, is this a tumor suppressor or an oncogene? It's an oncogene, right? Because you make a lot of it, it goes forward. What about this? Oncogene. Because if a lot of it's there, you saturate the system, it's going to go into mitosis. So anything that drives something into mitosis faster, more reliably, is an oncogene. Okay? Anything that's trying to put a brake on that is a tumor suppressor. So as we go through the cell cycle, we'll encounter more of those examples. So this is, it turns into a wee little cell with an Irish accent. Okay? That's where it gets its name. Okay, so this is going to be fully active. Now, the real question is, why does that cause mitosis? Certainly defeats the checkpoint, so this is now active, and that just summarizes this. Okay. So where we are is we're going from interphase into prophase. Okay, so how do we get from here to the next checkpoint is really the question. Something that mitotic CDK does causes that. And so this is a threonine-serine kinase, so it has to go around phosphorylating targets. So what is it that gets phosphorylated that makes it go through here? The nucleus has to break down, the chromosomes have to condense so that they're easily moved, and then microtubules have to depolymerize because they were all over the place, right? Now they have to decondense and reform as spindle fibers so they become very unstable. Okay, all right, so let's talk about those targets. This is a really obvious one. If your cytoskeleton is all over the place and you're very stable, you've got a Golgi and ER in very particular places, and you're trafficking things, this is very specific. Okay, so you're going to have to depolymerize all of that stuff and turn everybody into spindle fiber. To do that, this has to become very unstable, and the way you do that is you phosphorylate this microtubule binding domain, not the projection domain, but this domain. And if you put a negative charge on it, it no longer binds to the microtubule. So MAP4 dissociates, that releases this and allows it to be unstable, fall apart, be reformed very quickly into spindle fibers. So this one makes a lot of sense. Okay. Okay. Second thing, what's going to happen if we cause H1 to become part of the nucleosome complex? What does that do for us?
Causes DNA to condense.
To condense. It goes quickly from the beads on a string to 30 nanometer solenoid, and then that gets wrapped up. So you got to have this go in there. So this H1 gets phosphorylated, and so that's also a target. And Also, you need the help of this thing called condensins, and this is, we studied this a long time ago. This is that structural maintenance of chromosome protein that's in the DNA scaffolding that causes the shape of the chromosome. And one of the reasons it's located as a matrix protein, nuclear matrix protein, is that it captures the DNA like a little bracelet, okay? And it causes it to loop even further and further compact. So that tells you exactly what this is doing, condensing DNA so it can be moved, okay? And this thing works really well if certain proteins are phosphorylated. Okay, so it gets phosphorylated, and that phosphorylation helps this do its job and tighten up the chromosome. Okay, so this is all should be making sense. One thing we have to do is we have to be able to take the spindle fibers now that they're forming and have them contact the kinetochore at in the nucleus, but you can't get into the nucleus unless you break this membrane down. There's a real quick way to do that because we talked about the lamins, which are intermediate filaments. As long as you phosphorylate them, they fall apart. And if they fall apart, the nuclear lamina falls apart, it releases the membrane. The nuclear membrane breaks into little vesicles, and the nuclear envelope goes away. And so now you have no barrier between the spindle fibers reaching out and trying to grab the chromosomes and allow them to bind, okay? So we got to get rid of this, and DNA detaches from the envelope, and the envelope falls apart. And meanwhile, all the microtubules that form the matrix on which the Golgi and ER sits, those are falling apart. So that all turns into little vesicles, okay? So everything starts to dissipate, but it's still there, it's just in a different form, okay? And this one is really important. We really do not want to split this cell until the right moment. So we want to inhibit cytokinesis until anaphase. So we're not there yet, right? We have to cross another checkpoint to get there. So in this checkpoint, which was mitotic entry, this is a downstream event of downstream entry, inhibiting cytokinesis. And the way you do that is to phosphorylate this guy, which is one of those, remember there are light chains and heavy chains for myosin. Myosin is the contractile machinery, but we can control myosin by phosphorylating the light chains. There's actually places, if you phosphorylate it, it activates it, and there's inhibitory sites. We want an inhibitory site to be phosphorylated, and that's what the mitotic CDK does. It phosphorylates that light chain. That prevents that from happening. Okay, so we're in holding mode. Okay, until we pass the next checkpoint. Okay, so that makes a lot of sense. So we've got this thing being condensed. We've got these guys. The sister chromatids are held together, right? And so we do not want those to pull apart. And so there's actually an inhibitory protein called securin that we'll meet here in just a moment that is preventing a protease from splitting this down the seams. Okay? And so we were keeping that intact until we pass the next checkpoint. Okay, so that all makes, so let's go a little closer into this. This is the centromeric DNA. You remember that in this region there's a lot of heterochromatins. That's because it's going to recruit the kinetochore proteins and form this really amazing structure called the kinetochore. And so it's attached to the DNA, and then the microtubule comes and inserts inside of this kinetochore. Now notice, it actually leaves a little space for this to continue polymerizing. Because what happens is you got to get the chromosome from wherever you pick it up to go to the metaphase plane. And that you may need to push it instead of pull it, right? And so you have to be able to polymerize this, and then this little thing's going to slide as you grow more microtubule length, right? So we won't talk too much about the mechanics of moving those chromosomes, but you use molecular motors to get this to move in the right direction. Okay, so that's why the structure looks like this. And so it's grabbed a chromosome, and this is where we're going to try to move into the next stage. So we've completed all the downstream events since we entered mitosis. We now have to get through the next checkpoint by doing a couple of things. Okay, And so we're trying to get through the next checkpoint, which is mitotic exit, or it's also called the metaphase-anaphase transition, because that's what we're trying to do. All right, so what's holding the sister chromosomes together is they're wearing an arm bracelet, okay? And this bracelet is another structural maintenance of chromosome protein. They're called cohesins, and so they're causing the sister chromosomes to stick together. And if we break that, the tubules, the spindle fibers are pulling already. And all you have to do is split it and then they'll go. They'll go very quickly to the poles. So all we have to do is unhinge the sister chromosomes bracelet. Okay? And the way you do that is there's a protein that we've been holding in check called separase. It tells you exactly what it's going to do. It's going to unhinge that bracelet by proteolysis. It's going to destroy one of these little subunits, and it happens to be SEC1. Okay? And if we unlink this bracelet, what's going to happen? You can pull the two sister tids apart, chromosomes. And so this is being held in check by a protein called securin that I just mentioned to you. And here it is. There's securin. It's holding separase inactive until we get to this checkpoint. Now it's time to let it go. All right. So the protein that's going to do that is this protein here. It's called anaphase promoting complex / cyclosome. We can call it ABC, anaphase promoting complex. But you do need to memorize that name, APC, because we're not going to write that down. All right. So this is held inactive after mitotic entry until we get to mitotic exit. Okay. So if you activate this, you're going to complete one of the necessary requirements to get to the checkpoint. Okay, so how do you do it? Guess what? You phosphorylate it. And guess who phosphorylates you? The mitotic CDK. So it's going to phosphorylate this on some serine or threonine. And what that does is it recruits, getting back to the car analogy, it recruits a driver. It has no way of driving to where it's supposed to go to do its job, and so it's turned off. But if you have somebody at the wheel that's not texting, then this thing can bind to that phosphorylated subunit, and now it's got a guidance system. Here's the driver, sitting on his chair, doing very comfortable, and it's going to target it to go to this complex. And so, guess what? This is a ubiquitin ligase. And we're going to see a couple of examples of ubiquitin ligases. Let's see, it's not my explanation now. Let's go back. So, for right now, you know what happens to a protein if you put a ubiquitin, and then you put a ubiquitin on the ubiquitin, and you create a ubiquitin chain. What happens to it? It goes somewhere in the cytosol. To the proteasome, it's the shredder. Okay, the cytoplasmic shredder. Okay, so all you have to do is tag this guy with ubiquitin, and it has a little sequence called the destruction box with some conserved lysines, and this thing is going to take, it has the name ligase for a reason, it's going to take ubiquitin and ligate it to that lysine. Okay? And if we tag this, it will be destroyed, and this will now become released, and guess what? That active separase is going to go back here and unlock that bracelet by destroying it. So what's the activity of this? So this is a ubiquitin ligase. This is a driver. It's a targeting device. Okay. This is just a protein inhibitory protein. This is a protease, and of course you know NDK is a serine threonine ligase. So all got different kinds of things going on. So if you activate this, you're going to pull the sister chromosomes apart, and they're going to go to opposite poles. Okay, and that begins anaphase. And you are now in anaphase proper. Okay. All right, now, so how do we ensure that we're going to wait to do this to the right moment? The right moment means a couple of things. One is that the kinetochores all must be attached to spindle fibers. If we don't do that, we don't ensure that, we could leave a chromosome behind and have all kinds of genetic mutations, which cancer cells love, but we do not. Okay. So, separated, this is just a little cartoon. Let's actually zoom in on that because it's a little small. It's from a different textbook. But your book decided not to give you this explanation. So this is just another cartoon of what we just looked at. Okay, so Separates is being held inactive by securin. APC destroys securin like this. That releases separase. It cuts the bracelets, and then they pull apart. Okay, so that's okay. The other thing, the second thing, I said there were two things that had to happen. The other one is that the mitotic CDK needs to be degraded, so the process cannot go backwards. If we stop this from phosphorylating targets that it does, we'll prevent the reverse reaction. So this is why it's unidirectional. Proteolysis makes things go one direction. Okay? And understand, you do not destroy the engine, you destroy the cyclone. You destroy the cyclone. Okay? And that's enough, because it has to have a cyclone, or else that T-loop is permanently in the way. All right, so now the second part of this problem is how do we ensure that all the kinetochores are attached to spindle fibers? That didn't work out. Let's go back, sorry. Okay. So this is what we want to ensure happens, and yeah. Sorry. God. Try it again. There. Okay, so this is the story of Mad and Bub brothers. So these two brothers are actually doing something really important, and that is when there's no spindle fiber attached to the kinetochore, they go there and they stick to the chromosome in the centromeric region. Okay, and they form a little brotherly complex. Okay, the Mad and Bub brothers, and that complex then recruits and sequesters all the targeting device for APC that ligase. So you soak up all the CDC20, therefore it cannot bind to APC, it's getting to the driver's seat, right? If you're there, it must mean, so it's going to recruit CDC20, and all the CDC20 is stuck to the chromosome and not to APC. So it holds that CDC20 inactive. Remember, this is the driver, APC is the car, the ligase, and so without the driver, you have no way of targeting the ligase to where it's going. See, that's really dangerous if this were just allowed to go all over the place, because the APC would just go start, ligating ubiquitin needlessly, okay, for things and destroying things. But instead, it doesn't go anywhere. It's inactive. That's because this is having a little chat with the Mad and Bub brothers. Okay. Now, what happens is if the kinetochore binds, this thing, Mad and Bub, dissociate, and then they release CDC20 from their grip, their little T part. Okay? And so now the targeting device can hop on to APC, the anaphase promoting complex, and it drives it to secure it and targets it, right? And it's going to drive it over to the mitotic CDK cyclone complex to cut up the cyclone. So that's how this checkpoint is controlled to make sure all chromosomes are attached. Mad and Bub. Okay. Any questions about that? Yeah.
What did you say stimulates the Mad and Bub to let the CDC go?
The attachment of that microtubule complex to the kinetochore.
Oh.
Yeah, it just makes them dissociate. Okay. Let's go back to normal view. All right, so this is how your textbook describes it. It's a little less. Okay, so it really requires, so it's actually not CDC20 that tells it to go to the cyclin, the mitotic cyclin. It's actually a separate targeting device. The reason there's a separate targeting device for almost every ligase. Because it really, so if there's 1,000 proteins that need to be targeted for destruction, you need 1,000 targeting devices. And so this CDH1 does the same thing. When it latches on to the APC, it's going to drive it to a different location. And this location is the cyclin that's attached to the mitotic CDK. So to review, CDC20 is a driver, but it takes it to securin. CDH1 is a driver, but it takes APC to the mitotic cyclin. And if you destroy that, what's going to happen to the engine? It turns off, actually. All right, so just a little bit about, I'm not going to expect you to know all these details. This is from a different textbook. But just quickly, you need, this obviously has to be tightly controlled because you can destroy a lot of proteins needlessly. So you have to have several different components, E1, E2, E3. E3 is the important one as far as we're concerned because that's the ligase that actually takes the ubiquitin and transfers it to the protein. And then it takes ubiquitin and transfers it to ubiquitin. And then it takes ubiquitin and it makes a tail of ubiquitin. That's called polyubiquitination. And this is what targets this to the proteasome, which is the shredder. Okay? And the shredder is very conservative. It actually ejects the ubiquitin so it can be reused in this cycle. And then the rest of the protein, in this case, securin, or it could be the mitotic cyclin, they go through here and it chops them up into peptides and amino acids, and then they can be reused. All right, so that's the ubiquitin pathway. And so if you're sitting there at the metaphase-anaphase transition, and you've got all your kinetochores attached, and APC is going around destroying what it's supposed to, that's what happens very quickly. So mitosis goes very quickly. At the same time, this starts to separate. That's anaphase A, and anaphase B, we're not going to go into that detail. Anaphase B, the poles start to move apart. And so that makes the connection between those daughter cells even more tenuous. All you have to do is going to tighten that sarcomere belt. And so to do that, what do we have to do?
Phosphorylate the microfilaments that contract out.
Well, in the right place. But the first thing you got to do, remember there's an inhibitory phosphate that's sitting there that the mitotic CDK. Yeah, they put it on the myosin short chain, light chain. And so you got to have a phosphatase to clip that off. And that's no problem because there's dozens and dozens of phosphatase trying to nibble on this all the time. All you have to do is prevent it from being rephosphorylated. And you've done that because you just killed off the CDK cyclin, right? So if you can't rephosphorylate it, the phosphatases take over. And they strip off all those phosphates from the light chain, and then that contractile belt goes like that, squeezes off and pinches the two apart. That's cytokinesis. All right, questions about that? Okay. Good. So that brings us to the second thing in the cell cycle. Let's take a break here since it's a good transition. Okay, why don't we get back to the story here. So now we've got some tools to figure out how we're getting through the cell cycle. There's one more checkpoint that we're going to focus on, and that is restriction. And it's really important in cancer because it decides whether a cell can leave G0, where it's kind of just sitting there cycling, not going through the cell cycle, it's just humming into itself, and trying to get enough nutrients and appropriate things like growth factors to bind. And if it doesn't have either one of those, it's going to stay in G0 or it's going to differentiate and never go back into the cell cycle. So how do we pass restriction? And the word start is because it was discovered first in yeast, so the yeast people, you didn't really want to go to any yeast people over there. They call it start, but the mammalian people that do cancer call it restriction. Because if a cell can just get to that checkpoint really easily, your cancer cells won't get into wild. Okay. So let's look at a visual of this. So one of the questions that we could ask is, why are growth factors required to get past this checkpoint? So that's the first thing we want to answer. And growth factors come from a lot of things. Platelets release these that are in the site of damage. And so they're there to plug the hole, right, so you stop bleeding, but they also release growth factors so that you start repairing the hole that's been poked in you. Okay, so platelet-derived growth factor is a classic ligand for an RTK. So the PDF receptor is an RTK, and what platelet-derived growth factor does is it dimerizes together and it does the cross-phosphorylation, and you have a growth factor pathway, or this pathway that we're familiar with, the RAS pathway turns on, and RAS, of course, informs MAPK to turn on, and MAPK does a lot of stuff in the cytoplasm, but it also dimerizes and goes to the nucleus and phosphorylates transcription factors. And particularly, transcription factors that sit on this little blue thing. It's called a serum response element. So it's a piece of DNA, it's a control element that sits on a gene that's going to drive the cell into past restriction. But that's a little bit far away. What we have to do is turn on there's two types of early genes that turn on and get you to go into the cell cycle from G0. And the first set are the immediate set. In fact, turning these genes, that is, causing transcription, requires no protein synthesis whatsoever. What it requires is this guy phosphorylating things. That happens very quickly. So you don't have to go into the nucleus, turn on a gene, come back out, turn it into protein, then it goes and does something. It's turned on because this kinase quickly goes in here, phosphorylates the transcription factors that sit on the serum response elements, and then the immediate early genes are expressed right away. That's why they're called immediate early. Okay? What are examples of immediate early genes? Okay. That would be old friends of ours, June and Foss. Remember, they're leucine zipper DNA binding motifs. When you stick them together, they form a protein called AP1 that sits on these serum response elements. Another famous one is c-MYC. This one is particularly overexpressed in tumors, and so it would be therefore a oncogene, right? These are oncogenes. In fact, we use these all the time to study, like when you're doing a brain scan of somebody and trying to figure out which areas are becoming active in the brain. You look for C-FOS upregulation. Okay, so these are the immediate gene expression products that come out, that messenger RNA is the first thing you see. Now, of course, these guys are going to go where? These are not proteins yet. They have to go, the genes turned on, at least messenger RNA. Now, it has to go back into the cytoplasm and be translated into June and Foss proteins and c-MYC. Where do they need to go? Back through they have to have the import receptor and that whole jazz, so this is all sticking together now. Okay, it's got to go through the nuclear pore, and controlling that process is a way of controlling cell proliferation, isn't it? If you can stop that, you can keep them out there in the cytoplasm, and the cell will not go through cell division. Okay, so what do you need these early genes for to get to the delayed response genes? The reason this is delayed is obvious, right? You have to sit there and tap your foot while all these messenger RNAs are going out through the nuclear pore, return to the protein, they have to come back in, they got to go through the nuclear pore, and then they got to find the response element. That takes time. That's why these are delayed. Okay? So the very first class of there's two classes of delayed response genes. And to make it really confusing, the full name of this is really delayed early gene expression. They left that word out. These are both early gene expression transcripts, but this one is more delayed than that because this one depends on that. So what kind of gene is being expressed that's delayed? It's this. This is a G1 CDK. This gets you out of G0 and gets you into G1. That's why it's called G1 CDK. And there are particular kinases, if you want to look them up, in case you want something else to memorize today, you can look these up. Instead of calling it that, if you want to memorize all these different names over here, have fun, okay? But we're going to call it that, right? So you need this one to get further in this whole process. So one of the first set is the CDK cyclin, the G1 CDK complexes. What is it that it does that's so important? Well, what it does is it phosphorylates this protein right here. Okay? And this is called retinoblastoma. And as you might expect, that when it's mutated, it causes the cancer retinoblastoma. And so what it's doing is it's holding these two transcription factors. They work as a pair. Okay, this is E2F and DP1. Some textbooks don't even mention DP1, but it's a dimer. It's already dimerized, so it can go into the nucleus and do its job. Okay, but retinoblastoma is holding it inactive in the cytoplasm. Okay, and so what happens is when the G1 CDK phosphorylates retinoblastoma, it releases its dimeric pair. And so this is a tumor suppressor, right? And so the way you get cancer from messing with a tumor suppressor is you inactivate it, you don't stimulate it. So if you ask that question, does mitosis increase or decrease if this is overexpressed? What would be the answer? It decreases mitosis, so it's a tumor suppressor. Right? If you have more of it that's active, this is the inactivated state. What inactivated it? That first delayed response gene, which is the G1 CDK. How does it do that? You know what a CDK does. It's a serine-threonine kinase, so you know what's going to happen. It's going to phosphorylate, okay? And so this releases these two to go into the nucleus and sit on a control element, and these two turn on a series of genes. This codes for the second group of delayed response genes. Remember, CDK, the G1 CDK is the first group of the delayed, and the second group is what? Required for two things: going through restriction, and when you get into restriction, you're in what phase? If you leave G1, you're committed to DNA synthesis. You're in S, so you're going to need the proteins to duplicate DNA. So those are the things that you're going to do, and that's exactly what you do. Okay, so what happens is this is the first delayed response protein that's produced, G1 CDK. It gets rid of retinoblastoma. Instead, they just show it as a block, but this is actually E2F with this binding partner, DP1. And so they sit on the response element, and that causes S-phase gene transcription. And it also causes the G1S cyclin. That's the one that gets you through restriction. But you're also going to make the cyclins to get you through S-phase. So you're making both of those. And the other thing is that when this becomes active, it comes back and sits on response elements upstream from itself. And so it positively turns on more E2F transcription. So there's positive feedback here. And then look, you're making more CDKs. What are they going to do? They're going to go phosphorylate more retinoblastoma. And so this thing goes very, very fast. Right? Okay, so the outcome of this is if you've made these cyclins, and the specific names are cyclin E and cyclin A, but we'll call them G1/S cyclins. These are specifically required to get you through restriction. Okay? And so you get these active CDKs, they feed back, cause more retinoblastoma to release, and so you get more active E2F. E2F sits upstream from its own response element. You can show that by looking at this. So if this gene is E2F, E2F sits upstream from its own gene, which is a positive feedback loop. Okay. And so now you have all the things you need. You have the cyclins, you've got the CDKs, they were already there. They just were inactive because they didn't have a binding partner. And you've got the other things you've coded for are the DNA synthetic enzymes that you need to make DNA. When is that going to happen? In the next phase, S phase. So this makes perfect sense of what you're trying to do after you cross it. So if you can get past this step, you need both these two guys to do that. Okay, so this is the first delayed early gene set, and this is the second delayed early gene set. Okay. All right. Now, so we've gotten past that checkpoint, and we're not going to go through the details of how you get through S and the rest of the G2. There are different CDKs involved. One thing that I forgot to mention is not only, let's go back to the getting through the mitotic exit. One of the things that APC targets besides the mitotic CDKs is it will actually destroy these guys too. And what that does is it prevents re-replication of DNA. So that means the cell is not going to be able to go through because you've destroyed the cyclin for this and the cyclin for that, so the cell can't possibly go upstream. It can only go downstream. And remember, proteolysis is what creates the unidirectionality. If you destroy something, it can't go back there. Okay, so we've gotten through there. Now, all right, so this obviously is what happens during cancer. If you can get rid of retinoblastoma by mutating it, okay, you're going to get cancer. In fact, retinoblastoma is an inherited cancer because it happens. Most cancers are sporadic. That means they just happen, and they happen to people that get old. What about a child that has cancer? What does that imply to you? They haven't been around long enough to pick up a lot of mutations. So basically, what had to happen in their genome for that to happen? They had to carry some kind of allele that was already a mutation that predisposed them to cancer. And so what happens is if they carry one allele for a messed up retinoblastoma, all they got to do is have one more and game's over, right? And so a lot of childhood cancers indicate, or in young people in general, let's say young women with breast cancer. That's because they're a carrier for that type of cancer. That's not typical. That's a genetic cancer. Most cancer that happens to old people because it takes a series of mutations have to happen. Okay. Okay, so let's talk about how your body protects you against going through the cell cycle when you shouldn't. Okay. And let's ask the question, why should you stop the cell cycle? Well, what if you damage your DNA? Okay, and it doesn't, you can go to the dentist and then not wear that little lovely apron, and this will happen, I promise. You'll split your DNA, and depending on what type of damage there is, these two guys, the automatic teller machine and this other thing, they give you some money, and then you go to the store, and then you can check out. Okay, so this is check 1, check 2, this is ATM. We're not going to worry about when you use these. But basically, depending on the type of DNA damage, you recruit these kinases there. Okay? And what they do is they're going to phosphorylate P53. And P53 won the Molecule of the Year award by Science magazine several years ago because it's always involved it's almost always involved in cancer. P53 is a tumor suppressor. It's one of the main curators of your genome. It's the protector. Okay? And so when you get damaged, P53 is activated. How does that happen? It's not magic. These are kinases. And so these kinases activate the check 1, check 2 kinases, and then they phosphorylate P53. This is just the reverse of what just happened in that previous slide. Again, it's the P53 is being phosphorylated. It is the tumor suppressor, but it's also the transcription factor. It's not an inhibitor that gets phosphorylated. It's the transcription factor itself. That causes this thing, MDM2, which has been, what it's actually been doing, it's another one of these ligases. And if P53 isn't phosphorylated, it means your DNA is healthy. And so MDM2 is active, and it continuously destroys P53. So this is so potent that the cell does not want it active as long as you're okay. But if you're not okay, if you spent too much time under a sun lamp or out on the beach, this doesn't have to be X-rays, it can be UV, it can be carcinogens in the environment. If it damages your DNA, what will happen is these will phosphorylate P53 and unbind the ligase from it so it cannot be targeted to the proteasome. So the The status of a normal person is for this to be continually made and continually destroyed. So it's just continually cycling. And then when it's told to stop, you do that by phosphorylating p53. Now, there are some cases when MDM2 itself gets phosphorylated, that happens. It's okay, but this is the primary mechanism. So what? How does p53 protect you from undergoing cancer? All right. So what happens is it becomes active and it sits upstream from a couple of different genes. The gene that it sits upstream that directly stops the cell cycle in its tracks is called P21. So this gene encodes a protein called P21. Okay, that's probably because its molecular weight is 21K, and so that mRNA turns into this protein P21, and it is a cyclin-dependent kinase inhibitor, a CKI, and they call it a CDK inhibitor protein, but the class is CKI, cyclin-dependent kinase inhibitor. And what it does is it just grabs onto this and prevents it from doing its job. It will bind to the CDKs. If you bind to that one, what's going to happen to a cell that's in G1? It's going to arrest, right? What happens if you're in it? You're merrily replicating DNA and you screw up, right? And that informs p53 to come alive. It's going to stop this dead in its tracks and prevent it from going through S phase. It's going to make you sit there. Okay? So this is what causes cell cycle arrest. It doesn't kill you or the cell, but it says, let's wait until we can repair that. And it tries to repair the damage. So that's what cell cycle arrest is. It's giving you a chance to come back and fix things. Okay. Now, what happens if you can't fix things? So it's irreparably damaged. Okay. Well, let's talk about it. We'll get to that point. There's a different way to activate this. And so you know how dangerous these are, right? These are the immediate early genes that turn on the delayed genes. Okay. So if you activate these, you're going to go through the cell cycle. You're going to get through restriction. Right. So if a cell makes too much of c-Myc has got like a target on its back. And if that gets out of hand, the cell detects the change in concentration. And what happens is it will inhibit c-Myc. So excessive c-Myc production will cause a protein that's not related to the capital A, capital R, capital F, which is that assembly protein. This is a different ARF. It's A, little r, F. So this is different. Okay? And what this does is it creates more ARF, and it will actually bind to MD into and pull it off. Okay? And so it's actually a protein-protein interaction instead of phosphorylation. So if you hold this inactive, then P53 is stable and it can do its thing. Because remember, that is the ubiquitin ligase. If you inactivate it, things will go forward. So you'll make more P53 and you'll get cell cycle arrest. We already know how you do that, right? You activate P21. It's a CKI. How do you tell the cell that you're hopeless? That's pretty much what you're saying. You're hopeless. And so you need to undergo cell program death. And that's a very careful process. Okay? So this is the leg that we've talked about so far. You get, in both cases, you get P53. This is the one we talked about. But if you want to kill your cell, you turn on a set of proteins that are called pro-apoptotic. And it tells you exactly what it does. If you're pro-apoptotic, you're pro-death. If you're anti-apoptotic, you're preventing cell death. Okay? This causes cell number to decrease. This causes cell number to increase. Right? So anything that does this is a tumor suppressor. Anything that does this is a oncogene. Okay? So it's pretty straightforward. So the way Puma works is it's probably a big athletic shoe. So it's going to step on BCL. It's just going to step on it. And when it steps on the anti-apoptotic protein, it's preventing cell death. It's saying, well, you want to stay alive. And it stops that, then the cell kills itself. Okay, so the way that works is it's pro-apoptotic because it prevents the anti-apoptotic mechanism from doing its job, and so the cell goes ahead and commits suicide. Okay, so to summarize, there's two different ways that P53 works. It will arrest you dead in your tracks because it inhibits the CDKs, okay? But that doesn't kill you, just arrest you. If you want to kill the cell because of irreparable damage, you stimulate the production of pro-apoptotic proteins. And one of these is called PUMA, okay? And then that's going to inhibit the anti-apoptotic proteins that are standing in the way of cell suicide. It's saying, no, don't do it. PUMA steps on it, the cell goes around, goes ahead, and kills itself. Okay, that's a lot to digest. Questions off the top of your head? Okay. All right, so let's go back to the issue. Why is P53 such a large component of lots of cancers? Well, there's two reasons for that. It's central in the process of cell cycle arrest and cell death. If you can prevent those two things, cancer cell has got it made. There's not anything you can do. Okay? So that's one reason. It's central to cell death and cell cycle arrest. The other thing is it's easy to mutate. It comes in the form of a homotetramer. And, okay, so what? And it's got a really cool 9 beta strand, beta barrel DNA binding motif that we're not going to look at. It's a very unique motif to bind DNA and turn on P50, P21, and PUMA. Okay, but that's not what's going on here. If you inactivate one of these subunits, so all you got to do is inactivate one. That is, not both alleles, like retinoblastoma. One allele, if it's mutated, it causes the rest of this to be inactive. So that's bad news. You only have to take one hit and you're gone. So this is a really bad tumor suppressor because it doesn't do its job unless all 4 subunits are working. Okay, so just briefly a little bit about apoptosis. And then we get to a point where if you looked at this in advance, there's stuff that's test material and stuff that's not. Okay, so the next chapter we go into apoptosis, and so that's what we would normally do. If you take cell 2, that's what you will do. But I just want to give you just a little introduction. But it's really I left it with what's PUMA and what's this BCL thing. So what is apoptosis? It's this orderly cell death, and one of the first things that happens is the chromatin condenses, the nuclear envelope falls apart, and the cytoplasm starts to sort of shrink. And then pieces of the cytoplasm carrying off some of your important organelles bleb off. Okay? So it starts to just sort of, bleb. All right? And so what's happening is it's making nice little juicy pieces for lymphocytes to come along and eat. So all these little pieces will be taken up. You're not going to release inflammatory cytokines. So it's not about inflammation. It's about orderly destroying the cell that's messed up. Okay? And so this is what it looks like. Very distinctive. Okay, so there's a lot of detail here, and that's not the point of this. The point of this slide is to show you that there's 2 pathways to undergo cell death. Okay? And one of them is the one we're going to talk about, and that is if you stop having trophic factors, if you don't even have to mess up your DNA. If you take insulin and platelet-derived growth factor, and there's nothing there to stimulate you to go into G1, and now you know why the trophic factors are so important, right? You know that you got to activate MAPK to turn on the immediate early, and then the immediate early turns on the delay, and then the whole story. Okay, so if you have none of that, what's going to happen? You can't go into G1, and if you're not differentiating, you don't have those signals either. It means it's time for you to go away. And what happens is PUMA will be activated. And what PUMA does, in short, is that it actually recruits these anti-apoptotic proteins that are keeping you from killing yourself, not you, your mitochondria. Okay, so there's anti-apoptotic proteins are binding to each other to keep themselves inactive. And what Puma does is it actually recruits one of those anti-apoptotic proteins, and it causes the rest of the proteins to form a little pore that allows cytochrome C out of the mitochondria. Where is cytochrome C, by the way? We're going to come back and pick up the mitochondria, so if you don't know, don't feel too bad, but you know, it's in the electron transport chain. There's some burgeoning biochemists in here.
Peripheral inner membrane protein.
It's a peripheral membrane protein, and it's the inner mitochondrial membrane, right? Because obviously, if you pass the
outer
electrons,
side of the mitochondria.
right? So it's a peripheral protein, but it's too big to go through a pore and then get to the cytoplasm, correct? It's too big, so you have to form a special pore to eject cytochrome C away from the mitochondria and have it give a whole new, different role in the cell called death. It's a death messenger. Okay, so you should not be secreting cytochrome C because if it gets loose in the cytosol, it's going to kill the cell. Right? So Puma is going to interfere with this protector called BCL and hold on to it, and so it can no longer protect the cell. And the cell will then go, okay, I'll form these pores, and then it ejects cytochrome C, and then all this jiggery pokery happens, and the cell just starts chopping itself up with these enzymes called caspases. It comes from a term, they have C-terminal aspartate residues, but their job is to go around killing things, okay, and chopping them up. All right, so this is called the intrinsic pathway, because every cell has this pathway. Then there's the extrinsic pathway, okay? And that's if a immune cell, just a good example is if an activated killer T cell says, you're messed up, or you're a cancer cell, you look funny, you should die. And so it will come, bind to you and say, kill yourself. That is where this comes in. So actually, it'll be delivered by the killer T cell, project this protein and shake hands, but really what it's doing is going to punch you in the face. And so you activate this extrinsic pathway. And it does the same thing. It puts together these caspases, which then activate themselves and then go all over the place and cause apoptosis. So now you know why Puma is deadly to cells, but it's deadly in a good way because it's been informed by P53 that things are wrong. Something's really wrong. Okay. All right, so this is just a brief thing. You don't need to read chapter 18, but some of you may or may not be interested. And so this is not for test material, and this should be under PowerPoint. Okay. So I just want to tell you enough about this to get you informed. So these caspases are very dangerous, and so they're highly controlled. They're made as inactive pro-caspases. So they have extra pieces of protein that have to be clipped off. Okay? And not only that, you have initiator caspases and then executioners. So you actually have to set up a cascade, so that's another protection built in. You have to activate the caspase, and then you have to have the right assemblage to cause the executioner into the room, okay, with this big axe. Okay. But they all do the same thing with proteases. And so you have one initiator activated, that can go around, because it's an enzyme, and activate many executioners, and then they can go through and chop up all the stuff that needs to go away. Okay. So here's this killer lymphocyte, killer T cell, and it's got a ligand on its cell surface. That's the handshake. It's not really going to be a handshake. Okay, and so it's going to bind to the death receptor, aptly named. And when it does that, it goes, I want you to assemble an active set of initiator caspases. All right, so that makes pretty good sense. Once you get this whole thing set up, those initiators activate just a few executioners, and then they proliferate to the cell and chop things up. Okay? And so that's how, this is the extrinsic pathway. Now, to the one that we're kind of interested in, how did we really get this thing, how did we get something weird like cytochrome C to get out of, to cross the outer mitochondrial membrane, because it is too big to go through a pore, right? So when it goes, it's a death signal, and it's going to bind to a protein that's going to assemble this thing called an apoptosome. And that apoptosome is just going to go around and activate all these executioner caspase. It's a real machine. I mean, it looks like It's probably as big as a nuclear pore complex. But we're not going to worry about the details. All right. So this just shows that cytochrome C, when it's localized to the mitochondria, is fine. But if you treat a cell, it goes throughout the cytoplasm, and that's not good. Well, it is good if you're a messed up cell. So again, there's two flavors of proteins. There's these, this family up here, and these are anti-apoptotic. This is what's protecting you when your cells are just fine, thank you. Okay? They're protecting. The pro-apoptotic proteins are, there's two different flavors. There's BAX and BAC. But this one, you've run into a couple of members here. Remember BAD? What was BAD? So BAD is a pro-apoptotic protein. Pro-apoptotic protein. And so it's going to interact with these guys and prevent them from stopping apoptosis. So the reason BAD is bad is that it will cause the release of cytochrome C, and PUMA is the same thing. It's going to prevent these from keeping you from apoptosis. All right. So that's the rule. There's the inhibitors, and then there's things that stimulate cell death. And BAD and PUMA are in that class. And so is this little protein called BIM that I'll just briefly mention. Okay, so what happens is the protectors, as long as they're kept away from each other, as long as they're talking to each other and they're inhibited from creating this little pore complex, then they're fine. But what these guys do is they jump in there and interact with them so that they are not dispersed, they instead aggregate. So PUMA and all those guys cause the aggregation of the anti-apoptotic proteins into a pore that causes cytochrome C to escape into the cytosol, and then everything is downhill from there. The caspases become activated. Okay, and so what happens is this is an inactive intrinsic pathway. You have an active BCL. BCL is keeping these guys from forming a cluster in aggregate. So this is anti-apoptotic. So if you can get rid of this, then these guys will very happy to form an aggregate and talk amongst themselves and create a pore. We don't want them to unless the cell's damaged. So when the cell's damaged, PUMA comes over here and kicks this one in the teeth, and it's inhibited. These guys aggregate, form a pore, the cytochrome C is ejected, and the cell dies. Now remember what insulin does. In the insulin signaling pathway, we activate by a lot of jittery pokery, AKT, and AKT, what it does is it phosphorylates BAD. And what happens when you phosphorylate BAD is BAD cannot come over here and kick this out of the way. Instead, BAD is wrestling with another protein called 1433. As long as it's phosphorylated, it can't interact, and BCL will continue to keep these guys separated, right? Okay. And so, if you have an apoptotic stimulus, you have a cell of BH3, there's a class of these things, PUMA is one of those. PUMA and BAD do the same thing. They come over here and block BCL. That allows these to get into the corner and talk to each other and form a pore and then eject cytochrome C. Yeah?
So what is it that causes cytochrome C detention in the membrane?
It's peripheral. It's just there. It's a peripheral protein, and something that happens when this pore is created, there's a change in the environment. It's very easy to get rid of it. Yeah. So it detaches. That's not a great answer, but I don't know. I'll look that up. So we know how insulin keeps the cell from killing itself. It phosphorylates things like PUMA or PUMA's uncle, which is bad, and it keeps bad from being a bad boy and kicking the good one out of the way. So your cells never undergo apoptosis, and so that's a way of increasing cell number, right? Preventing cell death increases cell number. Stimulating mitosis increases cell number. What is going on in the insulin pathway that does that? That stimulates cell number, not inhibits, stimulates mitosis, not inhibits apoptosis. The result of both of those pathways is an increase in cell number. So insulin's doing both. It's blocking apoptosis, it's stimulating mitosis. What's the mechanism? by which it stimulates mitosis. You now know the full mechanism. It will be on the test on Tuesday, I promise. If you not start studying, you're screwed if you haven't. All right, let's reason it from ground zero. Insulin binds to an RTK. We just talked about an RTK, platelet-derived growth factor. Insulin is the same thing. What pathway in that series of signal transduction pathways is going to cause the cell to be able to cross restriction? Because insulin is a growth factor. Well, it autophosphorylates. It has phosphotyrosines. It actually recruits the Internal Revenue Service, sticks to it, IRS, and insulin receptors. Okay, it sticks to it, and that is a scaffolding protein, recruits GRIM and SOX and RAS and RAF and MEK and MAPK, and we just talked about what MAPK does. It phosphorylates proteins and transcription factors called CFOS and actually CMYC2 and CJUN. So that's the connection between insulin and this thing and all the rest of it. It's all connected. So insulin is stimulating the MAPK is what the answer to that question was. MAPK pathway is mitosis. You can think of the M as being mitosis. Okay? And it also prevents apoptosis because it blocks PUMA and BAD from doing this. All right. And so just as a side note, what we found in our studies of cutaneous and T-cell lymphoma was if we give these patients HDAC inhibitors, it wipes the cancer out. They go from being horribly deformed because it's the skin, the lymphocytes are in your skin, just horrible lesions, completely cleans them up. And then about 7 to 10 months later, they redevelop. Something escapes. So there's a resistance mechanism. So if you don't get every single one of those, then it's going to come back. So what you have to do is be able to block the resistance pathway at the same time you give the HDAC inhibitor. So we had to find what that resistance pathway is. And what we found, the first clue, was these tumors, these lymphoma cells, were overexpressing insulin receptors. And we went, aha, that's what it is. And so our first guess was it's AKT, because AKT is going to block apoptosis, and that's classic for a lot of lymphomas. And we looked for AKT, and AKT is not necessarily going to be upregulated, it's going to be hyperphosphorylated. If you know that pathway that's going to be on the test on Tuesday, that's what happens, okay? And so we looked for it, it was not activated at all, just normal. AKT was not the problem. So we went, well, there's not increased mitosis going on here. It's just cells are dying. They're not dying off by apoptosis. There's still mitosis, but it's not excessive. And so we said, well, it's probably not the MAPK pathway, but we'll look anyway. And so we did a genomic scan of all those proteins, and we found one that was overexpressed, and that was MEK. Okay. Not MAPK, it's MEK. So MEK was doing something on the side, had a little game on the side, instead of just stimulating mitosis, it was doing something else that kept these tumor cells alive, even though they were being slammed in the face with HDAC inhibitors. Okay. And what it turned out was MEK was phosphorylating BIM. Okay. So BIM is in the same family as PUMA and BAD, and so if MEK phosphorylates BIM binds to, instead of doing this, and kicking that out of the way and causing the cell to eject cytochrome C, BIM was being phosphorylated and it tangles with this inhibitor protein called 1433, because 1433 binds to these phosphorylation sites on BIM, PUMA, and BAD. And so BIM was being inactivated. That allowed these guys to never cooperate to form an aggregate, so that aggregate could not form because this protein was doing its job. So if you give, so the new treatment that we're proposing, and it's pretty far along the process for being approved as a phase 1 clinical trial, is you give the patient HDAC, and then you give them a MEK inhibitor, and that has worked. It completely killed all the cells in culture. So, all right.